In this video, we're going to discuss some techniques used to purify proteins. And by the end of this video, what I'd like you to be able to do is to list several ways a protein can be detected while trying to purify it, to describe the general strategy for purifying proteins, explain the mechanism by which salt precipitate proteins. I want you to be able to compare and contrast the different types of chromatography. I want you to be able to interpret protein purification data. And finally, I want you to compare and contrast SDS-PAGE, isoelectric focusing, and 2D electrophoresis. So, as a biochemist, oftentimes you want to, you want to study a protein, and therefore you need to purify that protein. You can't work with a protein when it's contaminated and in the context of lots and lots and lots of different proteins. Many of the things you need to do to it, it needs to be purified. In order to purify a protein, however, you have to have a way of seeing it. And I got that in quotes. Basically what I mean is you have to be able to somehow detect that protein and whether or not it's in a particular fraction. And there's lots of ways that you can detect proteins through enzyme activity assays, ligand binding assays. Basically, if you know what your protein binds to, you can detect it that way. Antibody binding assays, poison assays if the protein that you're using is a known poison, hormone assays. Here we have an example of a protein that you might be interested in, lactate dehydrogenase. Okay, that as a biochemist, perhaps you want to study this protein and you therefore want to purify it. Well, lactate dehydrogenase is an enzyme, and what it does is it catalyzes this reaction. It catalyzes the reduction of NAD plus to NADH by lactate. And here's the interesting thing. When NAD plus goes from being in its oxidized state into its reduced state, it gains in it picks up the property of absorbing light at 340 nanometers. And you can measure this with a spectrophotometer. So you can take a fraction that you think has your lactate dehydrogenase in, you can add to that lactate and NAD+, and you can calculate by looking at the absorbance at 340, you can calculate just how much enzyme is there by seeing how much NADH or NAD+ becomes NADH. Okay, so this is an assay that you can use to detect the presence of your protein if, for example, your protein was an enzyme. Okay, so the basic steps of protein purification include homogenization, centrifugation, precipitation, dialysis, and fractionation via column chromatography. Homogenization is that first step where you take your tissue or you take your complete organism, you take your origin of your protein and you grind it up in a buffer. This is going to be the part that is going to be the least expensive part to do most of the time. It's going to be the highest volume part. You're going to have up to liters of liquid, of buffer that you're using here. At that point, you're going to take that large volume and you're going to pop it into a centrifuge, and you're going to centrifuge, and depending upon the density of your protein will determine where within that centrifuge tube your protein will lie. Now, there's still going to be lots of other proteins that are contaminating your protein of interest, and so that's what steps 3, 4, and 5 are, and we're going to go into each of those in a little bit more detail now. So ammonia sulfate precipitation. This is one of the most common ways to precipitate your protein. So at this point, what you've done is you have your protein of interest, that's the blue here, and it is dissolved in a buffer that has just come out of the centrifuge. And it's dissolved, meaning that it is surrounded by water molecules. So your protein then is at least a little bit water soluble. Now, what precipitation does is you add a salt to it, this ammonium sulfate, which breaks down into these two ions here. Okay, this positively charged ion and this negatively charged ion. Now, these ions interact much better with water than your protein of interest. Okay, so what that means is all of the protein-water interactions are now being replaced by water-salt interactions. These proteins are being left out of all of those interactions. They're being excluded from those interactions. So via hydrophobic interactions, what starts happening are those proteins begin to clump together. And so depending on just how insoluble those proteins become will determine how much salt you need to add to your solution. And you can then put this into a centrifuge it, get rid of the supernatant, and keep that protein. Or if your protein is very, very, very soluble in water, then you can precipitate out all of the other proteins and keep your protein, which is solubilized within this solution. But the problem is, at this point, even though you have selected your protein away from many of the other proteins, you have a problem. Number one, it's still a little bit contaminated with proteins, and number two, it now has all of this salt there that needs to be removed. And that's the job of dialysis. So what happens here is you put your protein solution into a dialysis tube. You put that whole tube into a buffer here. And after a while, what happens is although your protein of interest can't pass through the tube, the salt can. And so after a while, after equilibrium has been reached between the salt concentration on the inside of the bag and on the outside of the bag, at that point, you're ready to go and you removed at least much of that salt from your protein solution. Okay, now you are at volumes where you can start to do more intricate types of purifications. And you would start doing a column chromatography. And there's lots of types of column chromatography that we're going to go into, but all of them have this in common. They all have a solid porous matrix. This is usually made up of these polymer beads, and they're referred to as the stationary phase. They're going to stay in this tube. And then you have the mobile phase. That's going to be your protein sample that you pour through this stationary phase. And over the course of time, so each one of these tubes represents a moment in time. So it's the same tube that we're just looking at snapshot 1, snapshot 2, snapshot 3, snapshot 4, snapshot 5 in time. What happens is as your protein sample begins to run through the stationary phase, that stationary phase, depending on what it's made up of, will start to interact with different proteins in different ways, such that some proteins will run a little bit more quickly through it, and some proteins will run more slowly through it. So as these proteins pass through that stationary phase, interacting in different ways with that stationary phase, they are going to be collected at different points in time. Right? So we will collect here, this is called effluent, the stuff that's running out. And if we collect it at time point, let's say time point 4, that's going to be a different set of proteins than when we collect it maybe at time point 6 or 10. Okay, so that's how we run this chromatography. We collect different fractions that run through the stationary phase at different points in time. And then we have to take each one of these fractions and run our enzyme assay or whatever assay we're using to detect our protein of interest. Which one of these fractions is our protein of interest in? Now there are several different types of chromatography, and each type is basically a different type of polymer bead that makes up that stationary phase. So here we have what's called gel filtration, or sometimes called size exclusion chromatography. And if we look closely, each one of these polymer beads, these big gray circles, have pores in them. And those pores are fairly small. So that means that when we pass proteins through the stationary phase, the smaller proteins get caught in the pores and they run very slowly through this. The larger the proteins, they can't get caught into those pores and they run more quickly through the solid phase. So that when you start to pull out the various fractions, the larger proteins are going to be in your earlier fractions, and the smaller proteins are going to appear in your later fractions. Okay? So depending on the size of your protein, you're either going to start looking earlier or a little bit later and start testing each one of those fractions to see where your protein is coming out. What that means is your protein is now just a little bit more purified than it was when it was before. Right? So before you did this test, your protein was mixed with these small guys, these medium guys, and then here's your protein of interest right here, these green things. And now you've isolated your protein away from these medium size and these small proteins. Your protein is a little bit more purified than when it was before running this chromatography column. Next, we have ion exchange chromatography. Now, this is a way to separate proteins based on their net charge. And the way it works is here we have our solid phase, these big gray balls or spheres here, and you can see that each one of them are negatively charged. So that when we run proteins through this, the proteins that have a net negative charge themselves, so large net negative charge or light red net negative charge, they aren't going to be able to stick to these the solid phase. They're going to pass through quite quickly. Whereas any proteins that have some sort of net positive charge with a small or a large net positive charge, they're going to stick to these beads, these resin negatively charged beads, and they're going to come out in later fractions. So again, depending upon the net charge of your protein will determine when it's going to, which fraction it's going to come out at. If your protein has a net negative charge, it's going to come out at earlier time points. If it has a net positive charge, it's going to come out at later points. And again, you've now basically purified your protein a little bit more away from other proteins. And that's the job of these chromatography columns. And finally, perhaps one that's the most complex, and that's called affinity chromatography. Now, this is dependent upon you knowing that your protein binds to something else, that binds to what's called a ligand. So let's take a look here. What we see is this, represented here in the image is a green sphere. That's your protein of interest. And you know before going into this that your protein of interest binds to this little red sphere. That's called a ligand. Anything that a protein binds to is called a ligand. And so we have this equilibrium that if you add ligand to a solution, your protein will bind it and you'll have this protein ligand complex. And so what we do is we take advantage of this and we engineer these polymer beads, so these big gray spheres that are covalently bound to the ligand that your protein is, that we know your protein binds to. And so that when we run that protein sample through the column that has all of these polymer beads, your protein of interest will bind to the ligand, which is bound to the beads. And so here's your protein, and it's going through that column, and as it does, it's passing these polymer beads that have the ligands attached to it. And your proteins of interest will attach themselves to that ligand, which is attached to the solid phase here, and they will stay here, where all of the other proteins, right, all of the other contaminating proteins, they don't bind to this red sphere. That's not a ligand for them, so they'll pass right through. Which means now what we have is a situation where your proteins are stuck to these beads in this column. We need to get them out. We need to release them from those beads. How do we do that? We flood the system with ligand that is not attached to these gray spheres. That's what we have right here. A solution of ligand is added to the column. This is the same ligand, these same spheres, red spheres, but they are ones that are free. They're not attached to these gray spheres. So what's going to happen is they're going to flood through here, and those proteins that were weakly interacting with these ligands here are going to also start interacting with the free ligands that'll allow them to drop off the beads and to then flow through the column and can be collected, okay, in a later fraction here. Let's take a look at the data that one would accumulate as one was purifying their protein. I want you very much to understand how to read this data. So let's go through it very carefully and understand what we're looking at. Here we start off with our cellular extract, what we get after homogenization. And then we go through these steps. And with each one, we measure several things. The first, what is the total volume of the fraction? And what you're going to see, and this includes everything, this is just the total volume of buffer that you have things in. And as I said way in the beginning, that first extract that you get from homogenization, that's going to be up to a couple liters. And then as we move on, right, it's going to get lower and lower and lower. Okay, we're going to lose fraction volume little by little by little. And then the next thing, each one of these fractions, we take a small bit of it and we measure the total amount of protein in that fraction. And this is not just your protein, this is all proteins. Basically, we're going to use that property of tyrosine and tryptophan, that absorbance property, to simply pop it into a spectrophotometer, read the absorbance, run an equation, and figure out how many milligrams of total protein we have. Again, not just the protein we're interested in, but all protein. And take a look at this trend. As we go, we have less and less and less total protein. Okay? Next, moving on. Now, the activity. Now we're looking at the amount of your protein of interest expressed in units of activity, which means the assay that we're using is an enzyme assay, which means this particular data is for the purification of some hypothetical enzyme. Right? So we know what its substrate is, and we have a way of measuring just how much of substrate it's converting into a product, and that is called a unit. Okay, so a unit of enzyme activity converts 1 micromolar of substrate in 1 minute. And so we take each one of these fractions, and we run this enzyme assay, and we see, okay, how much of this protein, right, how many units of activity are we seeing? Right, so each one of these steps here shows us how much of our protein we have. And finally, maybe the most important number here is a number that represents the fraction, essentially the proportion of total protein that is what we're looking at. So basically what that is, it is the units of enzyme, right? So what we're looking for divided by the total mass of protein, divided by everything. And that gives us the specific activity, the units of activity of our protein of interest divided by the total amount of protein in that fraction. And notice, take a look at this number. It increases. Take a look at this number, the activity. So just simply looking at the units of enzyme that we're interested in, as we go through these steps, it decreases. Starts at 100,000, goes to 45,000. Right? That's total activity. It's decreasing. Think about why that might be the case. Why is that the trend? Is that a trend that we would want to have if we had the choice? Why is it happening? And then we have specific activity. That's increasing. All right, let's think about that. Is that something we want increasing? Why is it increasing? So think about these two questions, and we will talk about that in lecture. All right, so now you have purified your protein quite a bit, where your fraction that you have has 15,000 units per milligram. That's pretty pure stuff. Let's see how pure it is. We're going to use a technique called SDS polyacrylamide gel electrophoresis, or SDS-PAGE. And that's a way to separate proteins by size. And now you have to have a fairly purified source of proteins in order to detect and see each one individually. So gel electrophoresis is a technique used to separate various types of molecules. You can use it to separate RNA. You can use it to separate DNA. You can use gel electrophoresis to separate proteins. And in this case, we're going to look at proteins. The basics are this. You have a gel that is either made up of acrylamide or agarose, and you then load that gel with your protein of interest, your protein sample of interest, or your DNA sample of interest. And then you run a current through it. You have it in buffer here, and you run a current through it. And those molecules are going to run through that gel, through the pores of that gel, and they're going to separate based on their size. The smaller ones are going to run faster, and the larger ones are going slowly. And so there's something different about proteins when compared to DNA that we have to take into account when running electrophoresis. And the first question is, when you run it through a gel, which direction will that protein run? Towards the positive pole or towards the negative pole? Think about proteins. Think about how they are different from DNA. And try to answer that question. I'll give you a second. All right, let's think about it. What do we know about DNA? Well, DNA has a net negative charge, which means DNA will always run towards the positive pole. Can we say that about proteins? Do we know that every single protein has a net negative charge? No. Different proteins have different net charges. So if you just slap a protein on a gel and run it, some of the proteins will run to the positive pole, some of them will run backwards towards the negative pole, and some of them maybe have a net charge of 0, won't run through the gel at all. So that's one problem we've got. We've got to do something to make all of the proteins have a net charge. Next, another question. When you compare proteins to DNA, DNA does not have, when you purify it, doesn't have much tertiary or secondary structure. It's pretty much just a linear chain. Let's take a look, though, at proteins. Protein A looks like this. Protein B looks like this. But we know that's not the native state of those proteins. We know that those proteins fold in a very specific way. Maybe protein B doesn't have much secondary or tertiary structure, so maybe it looks something like that. Maybe A is a very, very globular protein and folds something like that. Now, when we run these proteins through a gel, and we don't do anything special, which one of these proteins is going to run more quickly, be able to get more quickly through those pores? A or B? It's going to be A. A is going to be able to, even though it's technically the larger protein, because it's so compact in its native state, it's going to burn through that gel faster than B. And if we didn't know this about the difference in their native structure, we would think that A was the smaller protein because it ran through that gel more quickly. So we need to solve a problem. We need to make it so that those proteins run through the gel based on their length, based on their size, and not their shape. Which means we need to get those proteins, no matter what protein we're looking at, to take on the same basic shape. How do we do that? We take and we denature those proteins, such that now they are both the same shape. They're long linear chains, and now when they run through, the differences that we see on how fast they run through is not based on their shape, but based on their size. We have solved one problem. We know that we need to denature proteins to get them to run through the gel based on their size and not their shape. But now, how are we going to make proteins all have the same net charge? And the answer is through a molecule called SDS, sodium dodecyl sulfate. It's got this non-polar tail here. It's got a pretty big negative charge right there. We add that to the protein. We add that to the gel. And what that does is it binds to proteins and it gives them all a net negative charge. On top of that, it also helps with denaturing those proteins. So now when you run your proteins through this gel, this SDS gel, they are going to run towards the positive pole, and they are going to separate based on their size and not their shape. So if we were to run various fractions through an SDS-PAGE gel as we are purifying them, so 1 would be the crude extract, 2, 3, 4, and 5 would be various other steps of purification, and lane 6 then is our pure RNA polymerase. What we can see is 1, 2, 3, 4 bands, which means what we get from this data here is that RNA polymerase is a protein made of 1, 2, 3, 4 subunits. And we've got a pretty pure sample of that. Now, we can go separate proteins a different way. Instead of separating the proteins, our sample of proteins via size, we can separate them via their isoelectric point. So remember, the isoelectric point, the PI, is the pH at which a protein has no net charge. So what we do is we take a gel and we take that gel and we put a pH gradient. So up here we have a very high pH. Down here we have a very low pH. And then we take our protein that we haven't treated with SDS, right? This is a completely something different than the SDS-PAGE gel. And we put that sample of protein in there and we run a current through it. Okay. Now, way up here at the high pH, that means there's not very many protons. That means most proteins will have a net negative charge. And because of that, it's going to be drawn to the positive pole. So those proteins are going to be drawn through the gel towards the positive pole. And as it's drawn through this gel, remember it's decreasing pH. It's going to encounter more and more and more hydrogen ions, which means it's going to become more and more and more protonated. What is eventually going to happen? It's going to get to a point at which the pH of this gel is equal to the isoelectric point. It's going to get to a point where the net charge on that protein is 0, which means it will no longer travel towards the positive pole and it will stop. So at the end of running this, you're going to then stain it for proteins and you'll have these bands, and each band represents a set of proteins that all have the same isoelectric point. So knowing the isoelectric point of your protein not only will help you understand its net charge and various pHs, but it will allow you to separate them away from other proteins using this isoelectric focusing. Furthermore, you can now take this gel and you can turn it on its side and put it into an SDS polyacrylamide gel and now separate all of the proteins in this band have the same isoelectric point, but that doesn't necessarily mean they're the same size. So when you run it through this gel, what you're going to be able to do is separate those proteins based on their size. So now, when you look at this SDS-PAGE gel, it's got 2 dimensions to it. It's got a dimension from left to right here of increasing isoelectric point, and it's got this dimension here from top to bottom of decreasing molecular weight. This is, again, another way of really separating proteins, because it's possible then to cut out this gel right here and then purify the proteins away from that gel. You've got yourself a really purified batch of proteins when you do that. Okay, so as a reminder, I want you to be able to list several ways that you can detect your protein of interest while trying to purify it. I want you to describe the general strategy for purifying proteins. I want you to explain the mechanism by which salt precipitates proteins and how you can use that to help purify proteins. I want you to compare and contrast the different types of chromatography, of column chromatography. Be able to interpret protein purification data, look at each one of those pieces of data that you have to measure and what you can calculate. Compare and contrast SDS-PAGE, isoelectric focusing, and 2D electrophoresis.